Welcome to the Maine Science Podcast. I'm Kate Dickerson. Today's episode is a conversation I had with Dan Cortes, who is an associate research scientist in the Pera Lab at the Jackson Laboratory. Dan is originally from Mexico City, and hearing how he got to the point of being a researcher was a lot of fun. Spoiler, it includes a medical degree and a passion for helping people. Dan was a 5-Minute Genius speaker for the 2023 Maine Science Festival, and I've included a link to his talk in the show notes. If you watch it, it will provide some context about why he and I were talking about his last name. I hope you enjoy our conversation as much as I did. So Dan, welcome to the Maine Science Podcast. It is a delight to have you here. Your talk at 5-Minute Genius this year was so much fun. Like every year, I learned so much with just 5 minutes, but I really enjoyed how excited you were about it. But before I let you talk about that research in longer than 5 minutes, I won't cut you off this time. I would love to know how we got lucky enough to get you in Maine and your background. Like, what was it about science and research? When did you know that that's what you wanted to do?
I always knew that I wanted to be a scientist. And it's, of course, like not that I was 5 years old and I thought, yeah, I want to be a scientist, like with all the letters. But, you're curious about things and you don't know the name of it, but you know what it is. And I remember being a kid, as I said, like 5, 6 years old and watching documentaries on the TV. And I just couldn't blink. I just wanted to absorb it all. Those documentaries about how the body works. And I felt that it was so fascinating how, like, what inside of us and all those statistics that might not be really important, but they sound interesting. Like how many liters the heart pumps of every minute or something like that. And that got me into it. And I knew that I always wanted to be a scientist. And I was always like doing good in school because, as I said, I guess I was just a nerd. I loved school, but I wasn't a typical nerd. I did enjoy, like for today's standards, I would say that I would be not a good candidate for anything because I was still going outside, playing with my friends. I was born and raised in Mexico City, which is a big city, and it's just concrete everywhere. Well, that's not entirely true. We still have forests around. But in my neighborhood, we just played outside and the street, soccer, and just like any kid. So I still had fun. But as I was saying, for a kid nowadays, like you see the summer students applying these days, and they have done like a lot of things and a lot of awards, and like I wouldn't be able to be here. But yeah, I liked science and I was always doing good in school. And I always went to public schools in Mexico. And then I got into med school. And the system is a little bit different from what it is here. Like more like the UK system. Like you don't have to go to college. After high school, you go to the university. And I applied and I got into med school. And then after that, I worked as a physician, like a year, I want to say. But I knew that I wanted to be a scientist. So I wanted to do basic research and I didn't do much during med school. So I went back to get my PhD. And back then I was working already with stem cells, again, turning them into neurons, but I was studying Parkinson's. So what I was doing is I added a factor that makes the neurons survive better. And so they could artificially express that protein. So when transplanted into Parkinsonian rats, they could survive. So that's what I was doing. Back then I got my PhD and then I applied for a postdoctoral position in different places. I got into some job offers in Amsterdam and in Canada and here. And you asked, like, how did you get here? Well, I got here because I heard about Acadia National Park and that's what got me here. Like, I love nature. I'm always outside doing stuff. Like, I just couldn't resist it and I had to be here and I love it.
I totally agree with you about Acadia. I feel so fortunate that we get to live and we're not even next door to it like you are. I'm an hour away and I still feel like we've hit the lottery sometimes with the beauty around us to be able to go right to it.
Yeah, Maine is beautiful. It's not just Acadia. I think Acadia is just a sampler of it. But all Maine is beautiful. I think I always try to stress that out. Like, it's all Maine, just as beautiful. And yeah, like you say, like sometimes I'm in the middle of my work and I'm incubating something and I go for a run out into the park. I love trail running, so I can go up to the mountains, trail run, and an hour back. And an hour later, I'm back running experiments again.
Yeah, that's pretty great. I want to know, if you knew that you always wanted to do science, why med school?
Because I wanted to help people. I was always in Mexico involved in nonprofit organizations. So I really care about people. And I felt like that was the best combination of both. But later I realized that a way of helping more people would be if I could find something like a drug or like a mechanism, and that would have a higher impact. And that's why I decided to turn all my efforts into research. There's people that can see patients, like MD PhDs that can see patients and then do research. I am not that capable. I get distracted so easily. So I need to focus on something. And that's why I decided to let's just do pure basic research and then not see patients. That's how I abandoned my seeing patients.
Okay, abandoned is a strong word, I would imagine.
Yeah, to be honest, I miss it sometimes. But you cannot do everything in life, right? Like, as I said, I was a nerd in school and I loved physics and mathematics and other stuff, but you cannot do everything. Well, at least I don't have the capabilities to do everything. There's people that I'm surprised, like, how can they do a lot of stuff? I can't.
I think you're more normal. I think the people who can do everything are these remarkable people who can get by on 3 hours sleep.
Yeah.
So that's what it seems like to me. So you did Parkinson's research for your PhD, right? And then when you came to JAX, is that when you switched to looking at strokes? This is the time when you get to tell us all the cool stuff without the cowbell on what you're doing now.
So when I came here, Martin Pera, my boss, he had this project about studying this protein called TIRC1A. That protein is involved in all the different stages of neural differentiation and development. And it's also important it's being associated to autism and some other neurological syndromes. So he wanted me to do that project, and that's what I did. But then I had my own curiosities and I applied for an award and I got it. And with that award, I started this other project, the stroke project. So what I'm doing with that project is just using these stem cell derived neurons and cerebral organoids to analyze how the genetic background can affect the neuronal survival and trying to harness that knowledge for therapeutical discovery.
Okay, so can you break that down a little more? Are you trying to at a molecular level and then how you can model that or use that information? Is that kind of the idea?
Yeah. So for instance, I always use the example of COVID, right? That we just experienced that COVID, we saw people that died, like they got it really, really bad and they just died. And there were people that were just asymptomatic completely. And one of the main reasons behind that is their genetic composition. We are built differently. So what I'm doing is I'm trying to study that in vitro. And in a similar situation, these people that are resilient to COVID and people that are susceptible to COVID, I'm trying to understand that, but for a stroke. So what I do is I have a collection of different stem cells from different genetic backgrounds, from different people from Latin America, from Europe ancestry, from India, from the Philippines, from Africa, so on and so forth. And then I turn them into neurons and cerebral organoids. We can discuss later what a cerebral organoid is. And then when I got these differentiated cells, I developed this in vitro model to study stroke. And I applied the stroke model into these cells. And then what I see is what cell lines survive better, what cell lines die. And then what I did is, as you mentioned, I go to the molecular level. I go into the genes and see what are the force that is driven this susceptibility and this resilience. So I found some genes, some candidates that are apparently driving these differences. And now what I'm doing is, usually in science, what you do is you find something and then you have to prove it either by increasing the expression or decreasing the expression. Like in the susceptible cell line, what if in the susceptible cell line you express the molecules that are making the resilient cell line resilient? Or vice versa, like in the resilient cell line, you decrease those factors to make it behave as a susceptible one. So I'm conducting right now those experiments to prove that what I find is true.
So how did you even come up with the idea that there would be a genetic predisposition one way or the other for stroke? I mean, is that a standard thing at this point? It still seems so new to me, figuring out the genetic pieces.
It's kind of known that there are genetic differences among all of us. Like just look at our height, not all of us.
No, I totally get that. The part I don't get is how do you go from that to stroke as something that would have a genetic difference?
Yeah, well, I think it comes from two reasons. One of them is literature has shown that stroke and TBI, for that matter, traumatic brain injury, the outcome of patients is not the same after you adjust for age, gender, economical status, anything and everything. After you adjust for all that, there's still differences. So there's got to be a genetic component. And the other thing is that in the lab, we always see that the stem cells, when you try to differentiate them into neurons, not all of them do what they are supposed to do. So they do whatever they want. And that tells you that the genetic background is playing a role. So for that, actually, we had to develop a new protocol that can differentiate all different genetic backgrounds into neurons. So that's, again, telling you, like, when you want to make neurons, not all the cells are the same. There's some hidden to us pathways and signals that we still don't understand. But at least with this protocol that we developed, we managed to clarify a little bit of how to, regardless the genetic composition of the cells, turn them into neurons. And it's interesting because if you look into the literature, how to turn stem cells into neurons, there's many protocols, but all of them are only focused on a couple of cell lines. But when you try to do something else, there's nothing else. And I don't know if that's because people never looked into it or because people tried to and they couldn't. And they said, let's go back to what works. But we in the lab wanted to study those other genetic backgrounds because that was the idea behind the experiments. I study a broad genetic composition, but you cannot do it if you don't have a protocol that turns your cells into neurons. So that's the unforeseen thing that I was mentioning earlier. Like sometimes you have to plan experiments that you'd never thought of you were going to be doing. But we came up with this, and now we are studying stroke, as I was saying.
So how many different genetic lines do you have that you're working on?
So I'm working with 8 genetic different mouse cell lines and 10 human cell lines. They come from a broader bank, but it's hard to work with that many.
I was going to say that aren't most experiments maybe 1 or 2?
Yeah. And so you usually work with one, and then just to validate that it works somewhere else, you use another one, and that's it. Yeah, you call it a day. But the whole idea is that you want to study genetic differences. And for that, you actually wanted to go broad. And that's why we did 10.
There's a lot of interesting things about this. But one of the interesting things to me is from a general reader perspective, one of the main things that comes out of people who talk about genetic differences amongst different human beings is the number they throw out is like, we're 99% the same. Like there's hardly any variation. But yet there's like enough variation that it sounds like you can really find different attributes based on background, where people grew up, because of the lines of people that have been there this whole time. It seems like both are true at the same time, that we really are astonishingly all the same, but we can't dismiss the fact that there are these really interesting mutations that then follow specific lines of people that make huge impacts across the world.
I cannot agree more with what you said. I think both are true. And you forget about comparing us. Compare humans against yeast, if you want to. Many of the genes and proteins we have, we share them with yeast. There's people studying Parkinson in yeast. How crazy does that sound? And that's because some of the mechanisms of resiliency, or like something that is called autophagy, and they're shared between different species. So it's many components of our genome are still shared among many species. But also at the same time, it's also true that we are different. And why we are different as humans is mostly because regulatory mechanisms, the gene might be the same and the pathways are the same. And the genome doesn't change much. It's like cooking. Like somebody decides, like, I'm going to add more salt. Like you're still adding salt to whatever you're cooking. It's just that one decides to add a little bit more and the other one a little bit less. People have found out that the differences mostly rely on regulatory sequences sequences in the genome. So that's the difference. And there's this fact that I love, like in 15 years ago, I want to say, there was this study trying to understand genetic diversity in Mexico. And they discovered that if you compare just physically a person from West Europe, like Spain or Portugal, versus somebody from East Asia, Japanese, Chinese, they look totally different, right? And you can think you cannot get it more different. And then you compare two Mexicans, one from the north and one from the south. You might think, well, they're Mexicans. They might be just the same. Well, actually, the Eurasians are really, really close to each other, genetically speaking, and the Mexicans are way more different. There's a lot of reasons behind that, but my point I'm trying to make here is that it's really deceiving trying to understand genetic differences just by one or two traits, like the color of the skin or eyes or height. It's complicated.
I love the idea of two Mexicans are vastly more diverse from each other than the other ones.
Yeah. Because that's the thing, like what you see as a person is just a few traits, like what your eyes can see. But do you know how much albumin you're synthesizing at the moment? Do you know how much like your neurotransmitters your brain is secreting at the moment, or like how much ATP your myosin is binding in your muscles? Like you don't know that. And you're trying to describe to make groups of people based on what your eyes can see. It's ridiculous. It's just impossible.
It's a good reminder that there's a lot more going on. So the work that you're doing sounds to me very much like not early stages, but super basic research to just understand these different mechanisms. This is the question that I know a lot of research scientists really get annoyed with, especially coming from the culture of America of like, how do I apply it? Like, where's the application? So how many things are you discovering that will have some type of impact? Because you said you want to help people. Like, is this something that you think will happen in your research project, in your early lifetime of research? Will it take 25 years to really find enough that you can make a giant impact? Or is this one of those cool basic research things that you are finding out so much you can apply it right away?
Well, that's the thing. Like basic research sounds like something that people are just doing because it's interesting, but it might not sound like something you can use for a patient. But when I said that I wanted to do basic research, I meant, and that's how another way of how you define basic research is just to study the basic mechanisms of how things work. As a physician, that's why I always, every time I do something in the lab, I try to make sure that it's going to be applicable, maybe not in the near future, but at some point. This is relevant therapeutically speaking, because as I said, I found certain genes that are differentially expressed. One way of using that knowledge is trying to activate those genes, but that's complicated. So what I'm using now is just simple drugs to activate not the gene, but the pathway. And I see that by doing so, the neurons can survive better. So I think that that's applicable. The idea is that eventually the road is long. Like you find something in the lab, and from that point to it goes to a patient takes years. But it has potential because, as I said, like just by adding like a molecule that you eat, like you swallow for whatever, like hypertension or something like that, like just by adding that to the neurons, they make them survive better against the stroke insult. So there is potential. And as I said, I just need to prove that what I'm looking at is real. And then the next step would be testing these in animals.
We're going to assume that it's real and that testing animals happens. Is this something where the neuron survives better so that you can lessen the impact of a stroke after it happens kind of thing? I mean, is that your ultimate, that's what you're trying to get to?
Yeah. So the problem with stroke is that, first of all, the problem with the neurons in our brains is that they are really, really active and they demand a lot of oxygen and nutrients. And the moment you stop that flow, they cannot cope with it and they die. When a stroke hits, the neurons are going to start dying almost immediately. And then that is going to expand and expand and expand. What it's called the necrotic core, or just the dying center of it. There's not much you can do because by the time somebody arrives to the hospital, those cells are gone. It's on us to do something and make sure that that core is really, really small or not that much. So the next area around that is called the penumbra, which is just the area that is basically like the neurons that are a little bit sapped, like they are not doing well, but they can be recovered. So if you can help them, you might be able to recover them. And that's the issue with stroke. Like many of the treatments we have nowadays, we have one treatment, tissue plasminogen, but that's it. And not everybody is a good candidate for it. And that has been around for many decades and we don't have anything new. So I think we need to put some effort in it.
So why strokes? Why that in particular for you?
I think it's personal. My grandfather died of it and I relate to that. Yeah, it's personal. But as I said, I relate to people and it's not just me and my grandfather. It's like that made me think about everybody else, like people that died in that situation. And then it's a pretty sad picture. And it's like, I think about all the other families, right, that have suffered through that. I think that's why I wanted to study it.
I think that's great. It seems like some research, sometimes you don't see an application right away and you don't know what the result's going to be, but there's real value to me in connecting the dots and seeing something that you can really try to make an impact on and also advance really cool science in the midst of it.
Yeah, it's true. Like we have to push that forward, like trying to come up with applications to whatever we're doing. But also I cannot deny that basic research that seems purposeless is just as important. Like one of the biggest discoveries in science these days is CRISPR-Cas. And that came from, like, for those who don't know, it's just a way of editing the genome specifically to wherever you want the genome to be edited, like precisely. And that was research that was doing in bacteria. If I'm going to tell you, I want to study how bacteria can handle viruses when they get infected with. Like, why do I care? It turned out that we could use that knowledge. We are not that smart as humans, as we want to say. We always steal all the tricks that nature already came up with. And so that's the thing, like nature came up with an idea to find certain sequences when cells get infected with foreign DNA and then destroy it. And then somebody pretty smart, but not as smart as nature. And I came up with like, why don't we use that tool to find sequences in our genomes and just edit it? And that's a Nobel Prize.
So do you have a sense of how long is this? You got an award to do this project, not to put pressure on you, but does it have an end date? Or is it one of these things where it's a recognition that it's really important work that you have to just start on and they're not necessarily going to cut you off after, 2 years or something?
No, it does. Like all the grants and awards have a limit. And yeah, I'm reaching the end of it, but I'm about to publish what we have and then continue the research.
In the same line?
Yeah, I think so. Well, that's to be decided. I'm an associate researcher in the lab, and I'm still deciding what my future steps are going to be. If I'm going to stay here at JAX or move into another position. I still want to do similar stuff, like something that is translatable. But that's the thing with scientific careers. Like you ask somebody where that person is in 5 years, it might not be the same as before.
Yeah, it's really interesting. I'm not an academic, but I'm married to one. And it's a whole other world that it's not like going in industry or going into teaching explicitly. You really have to go where the jobs are and you have to find a match. And it becomes a tight-knit community, but it's one that is spread often across large distances. It's interesting to observe.
Yeah, it's like you need to make a lot of networking and collaborations, and that might be next door or across the country. Yeah.
So let me ask you this. This is a totally separate question from all of that. How difficult is it to be doing all this work in not your native language? Like when did English become the thing that you would have to rely on to do all this communication?
I think I'm going to ask you that question because I don't know, like, as long as you can understand me, I'm okay with it.
No, I can understand you, but at some point you had to switch from Spanish, right, into English. When was that?
Well, I studied English since I was a kid. I'm not going to argue that my English is perfect because once I finish a sentence, I realized that I made one or two mistakes.
Okay, but I guess my question is, I recently talked to Caitlin Howell, and she got her PhD in Germany. So she was part of 5 Minute Genius too. And she said she did not have to worry about learning German because English is now the language of science.
Yes.
Right. Which is great for Americans who are terrible at learning new languages. We can put that out there. We're not very good at it. So it seems to me if you don't have English as your native language, it's an extra burden to make sure that you're communicating well. So I'm just curious, like, I know you've been studying English for a long time because that's the way systems work outside of the States. You learn another language, but it's got to be different to do science at a high level that you're doing at. How much adjustment was there for that?
Yeah, it's hard. I think it was hard when I came here because you want to succeed, right? It's maybe not that I wasn't capable of. It's more about putting too much pressure on you because you think that you are not going to make it, that your English is not that good. You just put too much pressure on you, whether you are good or not. But that's not real. Like, as I said, as long as I'm able to convey my idea, I'm going to be fine. But the pressure is always there. And for us, as you said, anybody that is not their first language. When I was in med school, the books are cheaper in English than in Spanish because you still have to add a translation fee into the book. And I saw that like as an opportunity, like I'm going to save some money, but I'm also going to refine my English. And so it was a win-win situation.
That's actually pretty great. I'm not sure I would have picked studying in another language.
Yeah, it's not really, like you have to convince yourself because it's harder. And it's like, I'm like, studying medicine is not easy. And like, am I going to do this in English? It's going to be harder. So yeah, it's not, don't think better than, don't think that it was an easy choice. Yeah.
Yeah. Okay. That's fair. So last question for you. As a postdoc, which I assume people who listen to this podcast have gotten used to talking about, back before I understood how science worked, I was like, what? You have to do more studying and research after your PhD? What is wrong with these people? But part of the cool thing I've learned about postdocs is you're in this really great position of possibly changing your area of expertise, but also mentoring graduate students and younger scientists, and hopefully helping them avoid some of the pitfalls that you may have encountered. So I'm curious if you've been able to do that as a postdoc, if you've been able to mentor, and if so, what you thought of it.
All the time, we have a lot of, from all levels, from high schoolers to grad students, or even people that are my level or high level because they don't know about stem cells and they want to learn. So they come to us and then we teach them. Right now, today, we just got the new batch of summer students. So we're going to have one, and we have one every year. So yeah, we're always mentoring people, and it's a great experience. I do like it. It's hard. I'm not going to say that it's not, because you still have to do all your job and then teach somebody else. But I think I like to pass on my knowledge or whatever wisdom I have, like the do's and don'ts, so they don't do what I should have not done. Yeah, and I like that.
This is going along with your theme, Dan, of wanting to help people on a larger scale. This is great. I've gone through this whole conversation and not called you Dan Cortes, even though I wanted to.
Yeah. The card you gave me is on my fridge now.
Oh, good. I'm really glad you were able to carry that with you.
Yeah, I love the Cortex last name.
This has been great. I really appreciate this. I know I had to get squeezed into your schedule, so I'm really grateful for your time. I'm especially grateful for your work. I think it's really interesting and important. I marvel that you're doing basically 18 different genetic lines to figure things out. That's pretty awesome and intimidating. And I'm really glad that you're working on it and will save someone else from having to work on all of those. So I appreciate it. Thank you.
Yeah, thank you for having me.
Thanks for listening to the Maine Science Podcast. Make sure you never miss an episode by subscribing on your podcast platform of choice. And please leave a rating and review. It will help more people find us and help spread the word about some of the remarkable people doing science in Maine. The Maine Science Podcast is recorded at Discovery Studios at the Maine Discovery Museum in Bangor, Maine. The Maine Science Podcast is executive produced and hosted by me, Kate Dickerson, and edited and produced by Scott Loiselle. The Discover Maine theme was composed and performed by Nick Parker.